Okay. So, good morning, everybody.
Good morning, sir.
Good morning. Now, today we discuss the Marsilia. Okay. So, Marsilia, the division is the Philicophyta. Class is the Lepidosporangiopsida. Order is the Marsiliales. Family is the Marsiliaceae. Genus is the Marsilia. Okay. So, this is a sporophytic plant body. This is aquatic, either is amphibians in the habitat. They can grow in the water as well as on the terrestrial areas also. So the plants are adapted to the growth in the shallow water in the wet places, shaded places are they can grow. So that's why these is the amphibians we call as the amphibians. So some plants they can grow as the sporophytic development under the aquatic habitat. So they can go as the conspicuous plant of the Marsilia is the sporophytes. So we know that it is the aquatic fern. It is the hardly resemble as a fern-like structures are there. See here, the plant body is the fern-like structure. See here, this is a Marsilia. This one is a Marsilia plant. There. So, sorry. So this one is also aquatic plants are there. This is Marsilia. Okay. So which is designated into differentiated into stem, leaf, and roots are there. So in these species, stems are slender-like structure, branched and rhizomatous rhizomatous structures are there. These rhizomes are creeping with these indefinite growth and are they found growing on the just below the soil surface. So the stem is nothing but rhizome or stem we call it the rhizome. That is the stem is in the form of the rhizomes are there. That is the creeping like structures are there. They have the nodes and internodes also present. In the nodal region, they are the growth in the lower surface. Some rhizoids are developed. So upon the above the rhizoidal region, they grows a long slender-like structure that leaves are developed. The rhizome, it is the alternate case in the branch that are produced near below the leaves, but never from the axil of the leaf. So this is the very short and nodes and internodes are there. Another one is the roots. Each nodal one or to several advantageous roots are born on the ventral side of the rhizome. An exception is found in the some species are there, but in the minuta where the root arises the laterally, the growth as the lateral is also. Another one, the leaf structure. This the leaf structure is pinnately compound leaves are there. In the Marsilia, the leaves are alternate arranged on the two roots of the dorsal side of the rhizome. There are the petiole or the quadrifoliate compound leaves are the petiolated quadrifoliately compound leaves. So length of the petiole varies with the shallow and to found in the growth. The petioles are long, slender, like erect-like structures are there. So from the four leaflets, so two of the dorsal pair, which is the slanting higher, and the single leaf blade is divided into 4 leaflets. So there are the 3 successive dichotomizes are there. In the base of the veins of each leaf are dichotomously branched with the veins interconnection with the cross connections are there. So during the in night time, the leaf is fold upwards exhibitions in the succinate venation is there. Succinate venation is also there, and the base of the petiole, a small body is there. That is the small pea-like structures are there. That is the reproductive bodies are there. That one we call as a sporocarp. So here, see this one is the sporocarp. A born structures are there. This is the sporocarp. At the base of the petiole, this one is the sporocarp. Okay, clear? Are you okay? So now this is the sporocarps are there, and some advantages roots and rhizomes are there, stem and leaves are there. Okay. So the next one is the anatomy of the rhizome. So when we take the transverse section of the rhizome, which is the circular in structures are there. Circular structures are there, which is differentiated into 3 regions are there: epidermis, cortex, and steel. Epidermis, cortex, and steel is there. So epidermis is the outermost layer. We know that epidermis is the outermost layer, and a single layer epidermis is there, and some have the stomata is also there. And the cortex, which is a differentiation into outer cortex and middle cortex, inner cortex is also present. In the region of the next epidermis follows the outer cortex, which is the parenchyma tissue is there, and some trabeculus also there, air chambers also there in the outer cortex, means filled with the air chamber. So each air chamber is separated by a single layer parenchyma tissues are there, that is we call as a trabecule. So inner cortex is solid constructions are there, that is the some parenchyma stored in the reserve food material. So another one is the phloem, means steel is the amphiploic siphonosteel is there in the amphiploic siphonosteel. So rhizome of the marsilia, that is the amphiploic siphonosteel. What is the amphiploic siphonosteel means? See here. Amphiploic siphonosteel means that is the specific character is there. See here. Means
Hello?
see here. So this is the amphiploic siphonosteel is there in the rhizome of the marsilia. So what is the amphiploic siphonosteel? So means the two amphi means two. Siphonosteel, amphiploic siphonosteel is there. That is what is the amphiploic means the phloem. So that is the outer layer is still is the pericycle with a single layer rectangular cells are there. Outer pericycle and inner pericycle is present. The xylem is forms a ring surrounded by the layer of the phloem on the both sides. Means xylem is surrounded by the both sides of the phloem, outer phloem, inner phloem, and outer pericycle is also there, and endodermis is also there. So that's why the xylem is surrounded by the both sides, outer side and inner side, which is the layers are there. So that's why we call as amphiploid Siphon of Stairs. This is the specific condition is present in the Marsilia. Okay, clear? Means outer endodermis, then outer pericycle, outer phloem, then xylem. So after that one, from center size to the outer phloem, then inner phloem, inner pericycle, and endodermis. Centrally, it is there. That is the specific condition of the Marsilia rhizome. So another one is the anatomy of the petiole. The petiole is the circular in outline structure is there. It is consist on the epidermis with the single layer of the rectangular cells are there. Which is the cortex. After that one is the cortex is made up of is there. The cortex is the have the some air chambers also there, means outer cortex, inner cortex, and middle cortex. So outer cortex is the number of air chambers are consist. Each air chamber is divided a single layer tuberculous are present. And inner cortex is the compact of the parenchyma and some hypodermis is also there. Means sclerenchyma tissues are present. Cholenchyma, some sclerenchyma tissues are there. So the xylem is located in the middle. V-shaped structures are there. That is the petiole is the V-shaped structure. Petiole is there. Okay. So next one is the see here the diagram is the next one. See here. This is the transverse section of the rhizome. The red color is the xylem is there. The xylem is surrounded by the both sides. So phloem is also there. Okay. Next one is the reproductive structure of the Marsilia. So the Marsilia is reproduced in the vegetative reproduction as well as the sexual reproductions are there. So in the sexual reproduction means vegetative reproduction means that reproduction takes place in the resting bodies are developed. Some germinate into the plants, nice conditions, favorable conditions are there. Some resting tubers will develop. Another one is the asexual reproduction. Here, the Marsilia is reproduced with the means heterosporous conditions are there. So heterosporous means two types of the spores are developed, means microspores and megaspores. That's why this Marsilia we considered as a heterosporous. The microsporangia and megasporangia are respectively to produce the microspores and megaspores. So these both sporangias are together to form enclosed in the sorus. So number of sorus are enclosed in the special reproductive organ is there, that one we call as a sporocarp. See here, there is the Marsilia sporocarp which is present in the base of the petiole. of the plants. So see here, this is the transverse section of the vertical section and longitudinal section of the sporocarp of the Marsilia. Okay. So the sporocarp is the born lateral and the petiole towards the base of the adaxial sides, which is attached a small stalk that is called as a peduncle. The number of mode of arrangement sporocarp vary with the species. So depending on the species, they can bore. The structure of the sporocarp is complicated when the bifacial sporocarp split open along with the margins of the dorsal anterior to ventral sides. So it gets open into two halves are there. The wall of the sporocarp is hard, thick and highly resistant and is mainly responsible for the tart, not like a appearance on the sporocarp. The wall is the resistant, that is the sporocarp are around the 50 years in there. So here, the inner surface of the each half of the sporocarp well included storage is the extent to from the dorsal to the ventral side is present. The story on the opposite wall alternate with each other. The number of stories inside each sporocarp is varies in between. Means there are the two to 20 of the stories are present. So the development of the sporangia on the receptacle, which is the commencement, all the top and proceeds toward the base of the on either side. So such sorus is described as a graded sorus. Each sorus is enclosed by a thin, delicate layer called as the indusia. So, which is the Marsilia true indusium is present. The structure of the sporocarp is best revealed by the study of the section of the longitudinal section. We know that. So that is the longitudinal sections are there. So see here. This is the longitude means. See, this one is the sporocarp vertical section means longitudinal section or vertical section. So there are the separating dorsal and ventral portions are longitudinal sections. The wall of the sporocarp is thick, having three layers are there. The epidermis and two layers of the hypodermis are. Both the hypodermis layers are consist of radially elongated cells with a thick and thin cell walls in the outer and inner layers is respectively. In the longitudinal section of the sporocarp, spori are the cut across and seen in the transverse views. The section of the sporocarp proceeding through the dorsal and ventral surface of the transverse section, the structure of the wall is written in the same above. In the transverse section as cut into the wall and the present in the longitudinal view, the single sorus present in the either side is the display to the elongated receptacle with each megasporangia or microsporangia. The section of the sporocarp passing to parallel in the two halves are there in the wall of the tangential section. The complete length of the work you can see here. The morphology of the sporocarp means there are the unity opening regarding the morphology of the sporocarp. So many interpretations are there. So that is the microspores are haploid since they are formed in the spores or either result in the meiosis. The megasporangia, which is developed at the top of the receptacle in the middle, have a short stalks are there. The megasporangium also shown the single layered wall and double layer of the tapetum inside with only one megasporangium seen at maturity condition. In the megasporangium, also several spores are found in the microsporangium, but not only the one mature and other is the degenerated are there. The development of the microsporangium and megasporangium are the like almost upon the final stage, where both sporangiums are developed in the single superficial development, typically that are called as electrosporangium. So structure of the megaspores. In the microspore and megaspore, ellipsoidal and large, measuring around 0.45 millimicrons are there horizontally. The fall of the spore is having the two layer each endospores and expose are there. Next one is the gametophyte and sexual reproduction are there. The spores are commandments in the gametophytic generation. The microspores here some the microspores are developed within the after their liberation and development in the male gametophyte. Good morning. So this is the development of the microspores. So the megasporangium germinate within the 2 hours after their liberation, and mature female gametophytes are developed in 14 to 20 hours. The female gametophytes are also formed within the megasporangium. So the female gametophyte is formed from the small apical cells and development cutting the two phases are there. In the anticlinal division to form a single cell thick vegetation tissues. So in the female gametophyte, the inner central cells produce the single archosporium. So structure in which is simple. It is consist of a neck with a 4 rows of the cells having the two-celled height. The axial rows consist of egg cell at the bottom and above the ventral canal cells are there. And the single neck canals are present. The female gametophyte marsulia regard as highly reduced since in the development only one the archegonium. That will be in the at the female gametophyte is developed within the megaspore is surrounded by the ovoid gelatinous envelope with the inverted funnel shaped prostaceous towards the apex. This envelope is more watery and attached attract the spermatozoids with the gets stuck up. The spermatozoids swim down towards the archegonium, which is by the expected by the splitting of the megaspores wall and capillary region. The neck canal cells and ventral canal cells are degenerated, leaving the path of the through which these protozoites enter and each bottom of the archegonium. One of the spermatozoites fuse with egg and to form the diploid zoospore. Zoospore develop into an embryo that is ultimately form the young sporophyte. The substance R is not formed and the entire oospore develop into the embryo. And the four cells are formed. Two are the upper quadrate segments from form the stem, and while the lower part quadrate segments to form the root and shoot. So along with the development of the embryo, some lateral cells of gametophyte divide periclinally as well as the anticlinal give rise to calyptra of the thick layers are there. The leaf segment grows rapidly to consist of the embryo is developed. So this is the life cycle of the Marsilia. So alternation of the generation is there means
Hello, sir.
applied.
Hello.
Hello.
Hello, sir. Please speak loud, sir. We can't hear some words, sir. Some words
Okay.
are not audible, sir.
Okay. Okay. Some disturbance
Thank you,
are
sir.
there means some internet issues are. Okay. Okay. So this is the alternation of the generation is there. So alternation of generation means that the haploid stage and diploid stage means haploid gametophyte and diploid sporophyte generation. They are to each other. So that's why we call as alternation of generations. Next one is the stellar evolution in the pteridophyte. What are the stellar evolution? See here in the pteridophytes, the steels are muscular system is present from the pteridophytes only. Okay, so this is the steels are there means the phloem is there and phloem is surrounded to the xylem and that is there is no pith, that one we call as the protosteel. Okay, protosteel means stellar evolution, the older anatomical that will be Nageli and other supporting phylotic theories regarding the individual vascular stance of the leaf. They are pursuing the course of the leaf and axis as the structure and out of the vascular system of the pterophytes and some sonophytes also. So that is the when Tigam and delete the steel means the pillar-like structures are there. Steel is a Greek word, so which is means steel means pillar. So pillar-like structure, which is the central mass of the vascular tissues, with the with or without pith is present in the central. So some members are have the pith, and that is the parenchymal tissue is there. So based on the this is the pith is presence or absent. Here they are developed into the different types of the steels are there. So the stellar theory holds and body of the stem and root. They are alike with the basic structure. It consists of central cylinder, steel and surrounded by the cortex. So here the stellar evolution in the first plant in the muscular system have its region is the pteridophyte, which is also called the first vascular plant. You know that one means the vascular system is developed from this divisions only. That's why the first vascular plants called as the pteridophytes. In the evolution of simple and primitive vascular system, the steel is present in the pteridophytes as undergoes to the several progressive variation leading to the formation complex steels also there. So The evolution of steel is studied from the different aspects are there. That is the types of the primary xylem, composition of the xylem and phloem. Another one is the structure of the steel. So, what is the types of the primary xylem? Means there are the three types of the xylems are recognized based on the relation between the protoxylem and metaxylem. It is reference to their position. The xylem may be differentiated from the procambium from the point of the origin toward the center of the axis, means the center of the steel. And xylem is from the result to centripetal growth, that is periphery to center, that is called as the centripetal xylem. Means periphery to center region. That one we call as the centripetal xylem. In this, protoxylem is situated away from the center and the metaxylem toward the center of the axis, such as called as the exarch condition is there. So xylem may grow towards the point being towards the periphery of the axis. This resulting forming the centrifugal xylem. They have the protoxylem towards the center and the metaxylem away from the center, means periphery region. That one we call as endark xylem is there. In certain some plants are the centripetal and centrifugal xylem formed such that metaxylem occur around the protoxylem. This condition is called as a mesarch condition. So, another in the composition of the xylem and phloem. The xylem is complex tissues intended for the conducting the water and mineral substances are there. So, So in the pterophytes, the gymnosperms were certain expressions, xylem composed, the tracheids are allow in the certain other addition to tracheids, there are the three xylem parenchymas are there. The presence of tracheids in the xylem is the primitive feature is there. In advanced plants, tracheids fuse ends and to meet the walls and separating the cell walls dissolved from long tube cells are there, that is the vessels are trachees are called. The phloem is the complex tissue concerned with the conducting of the food substances. In pteridophytes, gymnosperm, the phloem is consists of only sieve cells and primitive characters are there. The presence of sieve tubes, companion cells, and foreign parenchyma is advanced character. So companion cells and pteridophytes, there are absent in gymnosperms. Another one is the structure of the steel. So this is the evolution of the steel is the being the structure of the steel in pteridophytes. It is traced from the different sources started. So that is a comparative study of the structure of the steel in the present day living genera from the main source. So another one is the study of the structure of the steel is possible from it is provide the comparative study, that is ancient and modern pteridophytes are there. First one is the protosteels are there. That is the protosteel, and during the evolution, the steel is pteridophyte shows a progressive variation. The protosteel to polycyclic dictyo steel with the translation stages of Siphon of Steel, solenoid and diffuse steel. What is the protosteel means? This is the simple and phylogenetically primitive type of the steel is there. See here, this diagram means this one is the protosteel is there. Means simple structure, xylem and phloem is there. That is the xylem and phloem is there. Here in the protosteel, it is consist a central solid core of the surrounded by the layer of the phloem. Okay. When the phloem is there, that is followed by the pericycle and medulla or pith is there. In the pith is absent in the protosteel. Okay.
Hello? Hello, sir.
So another one is the Siphon of Stairs. In the some species, that is the pterophyte, shows the protosteel in large, thin-walled parenchymatous cells may be associated with the xylem is there. In the central core of the uh, xylem is composed of the tracheids internet in the wall living parenchymatous cells are there. This is the earliest stage. A few parenchyma cells occur in the scattered in the xylem and core to form the mixed pith is there. See here, this is the xylem and phloem is there. This is the types of the stellar evolution means phloem and xylem is, and central pith is also present. So in the pith is surrounded by the ring of the xylem, a ring of the phloem and pericycle. Such modulated protosteel they appear as a hollow cylinder surrounded in the pith is described by the Jeffrey in 1905 as the siphon of steel. Siphon of steel are two types are there, that is the ectofluic and amphifluic. Siphon of steel, this is described into two types are there, that is the ectofluic, another one is the amphifluic. In ectofluic siphon of steel, the xylem cylinder is enclosed, the pith is surrounded by the cylinder of the phloem on the outer side only, with a pericycle and endodermis is outer side. In amphifluic means outer side, that is the ectofluic. In amphifluic means amphifluic siphon of steel. Previously we discussed that one. That is the steel is pith is may belongs to the lycopodia. That is the same primitive structures are there. Means both sides are there. The xylem is surrounded by the both sides mm, of the phloem is there. Phloem, outer phloem, inner phloem, outer pericycle, and outer endodermis. Like that structures are there. The another one is the solenosteal. The solenosteal means solenosteal. The cyphenosteal, which is referred as scattered leaf gaps, there are the is not overlapping each other, is described as the solenosteal. Okay. That is the so in the solenosteal maybe is ectofluic or amphifluic is also present, which is the considered the so amphifluic cyprosteel as a solenosteal, while the in Ames and Daniel is not a distinguished with the solenosteal from the cyprosteel. Another one is the dictyosteel. In many of the cyprosteel pterophytes, where the vertical shoot is the apex is the short, the leaves arise at the crowded manner is there. This is the result in the further complication of the steel where the several leaf gaps overlap with each other. So that is the steel is there. The polycyclic steel, another one in it is the ultimate is the complex complication of the steel organization in the pteridophytes. So development of number of separate steels one side and the other side, it is known as the polycyclic. So the polycyclic steel have two in the peridium, at aquatium there are three in the motonia. So another modification of the Siphon of steel in the formation of the each much advanced type steel is there, that is the U steel condition is present. In this vascular system, it consists of collateral and bilateral bundles with a leaf gaps are. U steel is characteristically the higher plants as the gymnosperms and dicotyledons of the angiosperms. So another one is the atactosteel. It is the most complex tissue found in monocotyledons, which is the consist of number of scattered vascular bundles are distributed in the conjunctive ground tissues are there. That is the atactosteel. Some evidences are to supporting these stellar theories. Advanced the protosteel is the most primitive and under the several variations leading to the evolution of the dicotyledon and polycyclic dictosteel is used. So polycyclic dictosteel, however, the stem apex is the cyclosteel to the possessed birds in a mature stem. They develop into the macrophyllus and the leaf gap are formed when the buds are removed frequently. The steel is left as a solonosteel or sometimes as a protosteel. So what is the aspects are there? That is the solid xylem core that one we call as a protosteel, and heterogeneous xylem with the or without pith is there. Central pith surrounded by the xylem rings are there. The interpretation of the steel by the leaf or by overlapping the leaf gaps, appearance of the internal phloem and endodermis also there. Another one is the heterospory and seed habit. Pteridophytes, they are mostly heterosporous, homosporous, where the sporophyte produces only one kind of the spores are called alike. That one called as the homosporous. These the phenomenon of the production of the two kinds of spores, that one we call as the heterospore. So among the modern living pterophytes, there are the heterospores, that is the Selaginella, Marsilia, and Salvinia. These are the heterosporous conditions are there. So origin of the heterosporous means the paleobotanical experiment and development evidences suggest that the heterosporous is the developing due to the distillation of the certain number of spores in the sporangium. As a result, these disintegration of the certain number of spores or remain spores such as survive in the sporangium have better nutrient filtration, natural growth, and specialized spore process. So, in the experimental evidences have shown the nutritional factors responsible for the origin of the heterospore. The Selaginella plant grows under the stable light have produced the sabale containing only one microsporangia because they are withstand reduce the photosynthesis in the marsilia. The temperature was loaded by the sprinkling cloud water spray in the marsilia plant was found in the all megaspores and large number of microspores present in the sporangium were killed. So in the homosporous form, the sex determination is tested there in the influence on the gametophytes during the formation of the sex organ. But in these cases, the heterosporous forms and the sex determination exert their influence on the spore mother cell. The influence of the sex determination is shifted from the gametophyte to the sporophyte. These small shift but the significant ones in the result in the origin of the heterosporous and vascular cryptogam. Okay. So another one is the seed habit means so manifestations of the heterospores means heterospores is marked by the combination of spores in two different sizes, smaller microspores, large megaspores are there. These microsporangium, the number of microspores produced in many of not numerous where the megaspore is limitation number. The usual number of megaspores 4R1 is there. In the Selaginella, that is the 4 megaspores are formed. So differentiation of the size spores are related in the distinction of the sex gametophyte, the germination of small microspore produced male gametophyte and megaspore produced female gametophyte. So these male gametophyte bear the antheridia, the male sex organs, and female gametophyte bear the archegonia. This is the female sex organs are there. The heterosporous through the unisexuality of the gametophyte and the sexuality is controlled by the preceding the diploid generation. So another effect heterospore is the related retention the megaspore with the issue of the sporophyte until after the fertilization. Okay. In Salacinella, the megaspore is not in the liberation immediately, but have retained with the megasporangium, where they developed into the female gametophyte, where the archegonia at maturity stage. This entire the female gametophyte is formed within the spore wall. The gametophyte, algae and bryophytes and to remove certain pteridophytes and independent are there. In the Selaginella, they are offered the several liberations related to the origin and evolution of the seed habit. After attaining so much, it is not a long step from the condition existing in some Selaginella species. So the megasporangium is provided with the protective integments are there, that is with the opening at the apex enter into the microspores. The megaspores formed after the meiosis developed into the female gametophyte. They are bear the archegonia. The female gametophyte formed entire with the megaspore. The microspore after their liberation and in microsporangia and by dispersed by the wind or they catch the fertilization drop to secretion at the tip of the megasporangium. After fertilization means the pollen tube, the microspore germinate and produce the pollen tube growth. They have the megasporangium absorb the food material. The spermatozoites is released released in the closed chamber. The archegonia is liquid in egg. It is there after form and embryo will be developed. This fertilization we call as a siphonotomy. Means the method which is the spermatozoites are transferred to the means of pollen tube and the spermatozoites reach the egg. That one fertilization we call as a siphonotomy. Okay. Okay. So thank you for thanking attending.